DNA, structure, function and discovery. Nucleic acids are the organic materials present in all organisms in the form of DNA or RNA. These nucleic acids are formed by the combination of nitrogenous bases, sugar molecules and phosphate groups that are linked by different bonds in a series of sequences. The DNA structure defines the basic genetic makeup of our body. In fact, it defines the genetic makeup of nearly all life on Earth. What is DNA? DNA is a group of molecules that is responsible for carrying and transmitting the hereditary materials or the genetic instructions from parents to offsprings. This is also true for viruses, as most of these entities have either RNA or DNA as their genetic material. For instance, some viruses may have RNA as their genetic material, while others have DNA as the genetic material. The human immunodeficiency virus, HIV, contains RNA, which is then converted into DNA after attaching itself to the host cell. Apart from being responsible for the inheritance of genetic information in all living beings, DNA also plays a crucial role in the production of proteins. Nuclear DNA is the DNA contained within the nucleus of every cell in a eukaryotic organism. It codes for the majority of the organism's genomes while the mitochondrial DNA and plastid DNA handles the rest. The DNA present in the mitochondria of the cell is termed mitochondrial DNA. It is inherited from the mother to the child. In humans, there are approximately 16,000 base pairs of mitochondrial DNA. Similarly, plastids have their own DNA, and they play an essential role in photosynthesis. Properties of DNA: two helical strands that are wound around the same axis make up DNA. DNA helices are classified as either right-handed or left-handed depending on the direction they are wound. However, right-handed helices DNA is the most stable and benchmark structure. The two helical chains are in opposition to one another. As a result, one strand extends from 5 foot to 3 and the other from 3 to 5. The strands are made of 4 types of nitrogenous bases, A, T, C, and G. These nitrogenous bases serve as genetic information stores and, as a result, encode the amino acids that form proteins. Physical and chemical properties of DNA solubility: DNA is polar in nature and thus soluble in water. Its highly charged phosphate sugar backbone gives it its polarity. However, in the presence of salt and alcohol, it is insoluble. Absorption at 260 nanometers, the DNA bases can absorb ultraviolet light. A spectrophotometer can measure this absorption. The amount of ultraviolet light absorbed increases with the order of the bases. For example, at 260 nanometers, a single-stranded DNA absorbs 1.37 units, whereas a double-stranded DNA absorbs 1.00 unit at 260 nanometers. Denaturation and renaturation: on heating, both strands denature, and on cooling, they can renature. The melting temperature, which varies depending on the precise DNA sequence, is the temperature at which these strands are permanently separable. In contrast to the region of higher concentration At, which is only bonded with 2 hydrogen bonds, the region of higher concentration of Cg has a higher melting temperature because these bases are bonded with 3 hydrogen bonds, which require more energy to break.